Okay, so hopefully you're okay with the lights off again. Okay. Works better for me, for my headache. So can we domesticate germs? Isn't the time that is or will be open soon? Have a while to do it. It's not that long. It's just I'm giving you time to fit it into your schedule. All right. But basically what it is you watch a video, a TED. Have you heard of TED Talks before? TED Talk videos? It's a TED Talk. It's a little bit older, but it's one of the few talks I've seen on that website where they approach this idea of communicable diseases. If we understand a little bit of the evolution and ecology of microbes, we could do a better job of nipping spread of pathogens and shortening the cycle and keeping people from dying. And so I think it's a great way to look at it. And I wish more people would think this way. It is, I think, maybe a 25-minute video, and then the what's it called, quiz. It's just questions I want you to work through, and some of it will be on them graded. Might be a couple that I have to hand grade. So you have some time to get that done, but that's for points. It's not extra credit. All right, so think of it as a homework. So it's kind of getting at the epidemiology section of this semester. You did a little bit in lab with the marbles and the handshaking and looking at chemicals, disinfectants, antiseptics, a little bit on antibiotic profiles. And now with this idea, that will also kind of go into that realm of epidemiology, I guess. Questions on that? So put it on your calendar. Do not forget. It's points for sure, but it's all open book, open note. So then tomorrow is your lab exam, correct? So we want to put it in a separate room so that when you're done, you can go to the lab if you need to work on your group project. So it's going to be down in the main bottom floor of Weston Hall, room 1201. I think it might be labeled the anatomy lab. It's one of the human anatomy labs. So you'll go there to take the exam, and then the normal lab room will be unlocked and you'll be able to get in there. Questions on that? I don't know if Duncan or Jackie has given you any feedback, but this third exam will not be as long as the first two, and not worth as many points. It's a shorter one. It's more like a robust quiz, so I don't think it'll take you that long. Okay. I'm still working on grading the essay portion of the third exam. That's going to take me a little while yet. But if you want to go over the multiple choice portion in more detail, I can certainly set up an appointment with me and we can do that. Average was good, 77%. That was nice to see. The final exam is going to start with the virus material, and then we'll see what else. Viruses, human microbe, and I'm not sure what else. We'll see what we can get to. I'll keep you posted on that, obviously, as we work through this last bit. So I think we left off here with the idea that obesity is kind of a worldwide problem, and it's a very complex reason for it. There's many reasons for it, and so trying to tease all that apart. But the evidence is piling up that the microbiome can play a large factor in that, and they can show that manipulating the microbiome in mice studies and rat studies can make a huge difference. And so this has really got the scientists excited. I like that bullet point that we looked at last time. The nutrient value of food is more so influenced by the host bacterial digestive capacity rather than static caloric value. I think this goes a long way to thinking about why so many diets fail, or they work for some people and not for others. And I remember, you all have unique microbiomes. You don't have it exactly the same. You have, pretty much those four phyla we talked about. You have a lot of members that go into those four phyla, but when you get down into the species level, you are unique when it comes to your microbiome. So the other way to look at this, I think, is nicely put here: the metabolic dexterity of the microbes allows them to digest foods that our cells can't do the same way. And so we have certain limitations in our physiological pathways where we can't work with certain foods that we ingest, but the microbes can. So again, just because, you know, we'll get more into that when we look at fiber, for example. There's certain fibers that we can't handle digesting, but microbes can. One of the important concepts that's really showing up in a lot of the studies is they're looking at what types of microbes are present and what does that have impact of the short chain fatty acid ratios and types. So I do want you to know that phrase, short chain fatty acids, SCFA. We're going to talk about this a few times here. So, what they're finding is that the amounts and ratios of these short chain fatty acids can play a role in both the obesity state as well as the lean state. And now there's some, and I'll go into this a little bit more, but basically we're looking at these microbes when they eat the food that you eat. Some of them will produce these different short chain fatty acids. And this is something that is done primarily through fermentation. When they go through their fermentation pathways, these are some of the products that they're making. So this is actually from your book, and they are trying to show the epithelial cells of the gastrointestinal tract, and then they're implying here that you have members of the Firmicutes and Bacteroidetes phylum. And here they're zooming in on Bacteroidetes, theta, iota, omicron, and then on the Nanobrevibacter. I'll come back to those. Then here you're looking at this idea that as these microbes eat the food you eat, so they are trying to get their carbon, energy, these fat, electron sources from the foods that you ingest. And if they have to do fermentation in order to get that to happen, that's what they'll do. So these products, butyrate, propionate, acetate, those are fermentation products produced by the microbes. Those are short chain fatty acids. Okay, they're short chain fatty acids. Anything in science with an H on the end, it's an acid. Okay, so short chain fatty acids. What they're finding in studies, this is data from an earlier study, they're finding that depending on what short chain fatty acid you're looking at, it could either lead to obesity or not. So let's look at this a little bit closer. You have here the idea of propionate is, you follow that arrow down, you can see it's kind of complicated. What they're showing is that in some cases, if you follow the red arrows down, it can be a negative thing. It can lead to inflammation. But in other cases, with the black arrows zooming off to the side here, they're showing that it can actually be a good thing. It can limit hunger. It can decrease fat production. So that one's a little murkier, and some subsequent studies are showing that to be true. Acetate here, what they showed in this study, is not such a good thing. If you have a buildup of acetate by your microbes doing their thing, in this study they were showing cholesterol and fat increases, basically fat storage going on. So pro-obesity state for the host. Then if you look over to butyrate, butyrate's turning out to be kind of a star in terms of the short chain fatty acid profile. Butyrate seems to be a good thing. Over and over, we're seeing that butyrate is leading to what we want, which is limiting hunger and decreasing fat production. Some companies are even looking at producing butyrate and putting them in pills like supplements, so you can just take them as supplements. I don't know about that yet. into that. That's how geeked up they are about this. They're willing to do that kind of business. Okay, so let's see, what was the other thing I was going to say? Oh, inflammation. So that may sound weird. We're not talking about the inflammation that you get if you have a paper cut or a cut on your finger, and then your finger swells up and it's all tender and painful and takes a while for that to go away. That's inflammation. We're talking about internal inflammation, where you have, and it's usually systemic, often starting in the gastrointestinal tract, but then going systemically. And so it's this chronic low state inflammation. And if you go to the doctor and they're worried you might have something going on where inflammation is playing a role, they can do a blood test and look for inflammatory markers. And that's something that's routinely done. So this is, in general, and again, many, many studies are showing this, the data is piling up, that which microbes are there in your gut primarily, what are they doing, what products are they producing, and some of them can lead to inflammation, and that's not a good thing. You want to minimize inflammation. Inflammation can be a good thing, like with the paper cut idea. When you get a paper cut on your finger, that inflammation is your immune system delivering good stuff to that wound to heal the wound, and that's good, and it's temporary. We're talking about chronic, low-level systemic internal inflammation, and that's not good. Let's go back to these two, Methanobrevibacter up in the upper right corner and Bacteroidetes. So you're seeing here that in this particular study, Methanobrevibacter, they're saying that it's altering the transcriptome, which just means that it's influencing gene expression of Bacteroidetes. So this one is influencing Methanobrevibacter is messing around with the Bacteroidetes gene expression. And when they looked at that a little bit more closely, so here this is some data taken from people who have gone through gastric bypass surgery, and they're finding that with this particular study, that some of the archaea type bacteria that could be in your microbiome, it looks like if you have too many of those, it could trend toward obesity. And so with this study, we're looking at the gastric bypass patient. Here's the last set of 3 bars. And you can see normal weight on the first 3 bars. And in the middle is the obese patient. And so you can see the shift after the bypass. You can see that there's a great reduction after the bypass surgery was done. So they've been looking at these types of ideas too, more carefully with some of the interventions that can be done. But we certainly, there's many of us that have different archaea type microbes in our gastrointestinal tract. It's not unheard of. So with these two though, when they did this particular study, what they found is that when they put these either singly into germ-free mice or put these 2 microbes together and then got them into the mice, and then they had the mice either living separately or colonized them together. And what they were finding is that with Methanobrevibacter, it was altering the gene expression of Bacteroides. If Methanobrevibacter wasn't there, you see a different gene expression pattern in the Bacteroides. And what they found is that because of the presence of Methanobrevibacter, it was causing Bacteroides to make more acetate, which we know in this study, they're like, yeah, acetate's not such a great idea to have a lot of that, because in this study, it did increase fat levels in mice. And so what they were finding is that this was through the process of quorum sensing. We had quorum sensing going on where there were chemical signals being produced by the microbe form sensing signals, and that shifted Bacteroides. And so this was a very clear example of how, in this case, form sensing between microbes was contributing to fat deposition in the mice. So lots of work left to do on this idea of species interactions in the gut. There's, I mean, it's kind of mind-boggling to think about. And I think they're going to need to get into machine learning and artificial intelligence if they really want to get at this in finer detail. Here's another published paper where they were looking at shifting diets in the people. And so this was, how did they, is this the one? Oh, no, this isn't the one. So here, this is primarily plant-based diet versus animal-based. And then they track over time DCA, chemical that is implicated in mutating DNA. So DCA is often considered negative thing. You don't want to have achieved animal-based diet or bile acid too can be an issue. Have too much of that for too long. These animal-based diets. And so looking at, again, bile cell hydrolases, the fact that it's so high in nutrient-based diet is considered, and then the sulfite. You don't want a lot of that going on either in your gastrointestinal tract. And so they were trying to get at, you know, what kind of diet might be better for the overall health. They were doing bile cell hydrolases. So primarily because bile acids, they are linked to inflammation. So this idea that you can get, and in this particular study, one we just looked at, they noticed this microbe, Bilophila 1 morphia, and they're showing that, I think it was just looking at a paper where that one could also be implicated in inflammatory IBS, inflammatory bowel syndrome, IBS, might show up in patients with IBS. So this is a concern. If Bilophila is doing its thing, what tends to happen is it's one of the products it makes during its physiology is hydrogen sulfide gas, which is toxic to any living organism. You don't want to be exposed to high levels of H2S. And so that's inflammatory. And then bile acid too. In this particular study, they were looking at mice being fed milk with fat in it. And so they're saying if you follow a high-fat diet, that this could happen if you have Bilophila in your microbiome. And if I'm not mistaken, isn't the ketogenic diet, isn't that high fat? Pretty sure that's a high fat diet. And I don't know a ton about that diet, but I know some people I've heard, acquaintances and friends say, oh yeah, I went on the ketogenic diet and I lost weight. It was really effective. But I looked into it a little bit and it's like, I think the impact it can have on your microbiome is not worth it, in my opinion. And so it puts you into ketogenesis too, which I don't know if you want to do that long term. So that's a diet I would certainly myself never try, but you really do need to think about what choices you're making because it can have a profound influence on your microbiome and all of their activities and the kinds of chemicals that they excrete through their physiological pathways, all the products they make. And we'll also learn that the microbes can directly influence your gene expression. So you're promoting, depending on what food you eat, you're allowing certain microbes to, be in fairly high populations in your gut, and you can manipulate that ratio where if you choose, high fiber foods, then you get microbes that are really good at eating high fiber. and they will start to bulk up, their population size will go up and up. On the other hand, if you flip into low fat, low carb, or high fat, low carb, you will get a different ratio in population levels of very different microbes. And they're finding that that makes a huge difference. This is pretty in your face, but it's pretty much accurate that every bite you take, it's either fighting disease or feeding it. And that's what they're starting to come to. And when they look at the impact of the microbiome on your overall health, and not just the health of your stomach. Like we're not just talking like colon cancer or no colon cancer. We're talking about full body, all kinds of options for different diseases that could result. So this is gaining interest. Diet-induced extinctions in the gut compound over generation. What they're finding is that, especially with our Western diet, which is low in fiber typically, that you can kind of pass this trait on to your offspring, and it can compound over generations. So what your great grandparents, and then your great grandparents, parents, that all is influencing over the generation what they ate and what that did to the microbiome populations, the physical line. So they're looking at this idea and realizing that we're losing certain key microbes that we used to have back before industrialization and our rather sad Western diet. And they're saying even if you flip into like the best diet you can think of, it may not be enough to get back the microbes that seem to have gone extinct. And so there's this new term like rewilding the microbiome, where they're saying we may have to create like probiotics that have these strains that have seemed to have gone extinct, especially in the Western population. And even when they study the Hadza tribes, and because they're the most hunter-gatherer type group of people, and so they've been very interested in studying the Hadza microbiome, and even their microbiome has shifted where they've lost some key microbes that we see in earlier studies. So this is something that they're looking at. And then, so here, this idea of, it can, what you eat, if you choose to have, especially if you're female, if you choose to have children at some point, what you eat can influence the child even before you conceive, because it's your microbiome you're altering and influencing, but then of course you're going to house that fetus in you, and there's chance of the interaction between the microbiome to the fetus. And so this particular study, they were looking at prebiotics. You know what prebiotics are? The probiotic, it's the microbes, right? Prebiotic is food you eat that feeds the microbes directly. It's not really something you necessarily can use yourself, but your microbes. And so it's a lot of fibers, essentially, a lot of different fibers. So during nursing, they're saying, as well as there's studies showing that while the fetus is developing, as well as shortly after birth, what the mom is eating can definitely have it. Even looking at behavior, not just the overall physical health of the infant, but the behavior patterns. So brain development, that's a pretty profound statement. So if you're wondering, these are some examples of prebiotic foods. Even chocolate made the list. I love that. So these have certain types of complex carbohydrates that qualify as prebiotics. That means that those complex carbs are very good at feeding the microbes. Why do you think it says underripe bananas? You know, when bananas are more green than they are, freckly, yellow and freckly. Have you tasted the difference between a slightly green and a freckly banana? A freckly banana is way sweeter. And so if you eat bananas that are slightly underripe, you get more complex carbs. So that's why we're saying don't eat the freckly ones. Eat the slightly underripe ones for more of the complex carbohydrate. By the time they're freckly, what's happened is in that banana over time, as they go freckly, they go from complex carbs to simple sugars. They've broken down into simple sugars. And so you instantly get that. And then I just saw this the other day, prebiotics, the inulin, that's a good one. I try to look for that in a lot of ingredients. Inulin ameliorates SARS-CoV-2 infection in hamsters by modulating the gut microbiome. So they are actually finally starting to look at this idea of, can the gut microbiome influence whether or not you get COVID, and how severe will that COVID be? And they have noticed that some people get COVID and they're, it's an annoyance, but it's not that big a deal. Others, of course, die from it. And so they're starting to look at, well, what's going on in the microbiome of these people. And they are finding that people who had a high inulin intake that correlated with low rates of COVID infection and certainly surviving. So they're starting to look at this a little bit more carefully. I think that's great that they're starting to piece that together. So probiotic, prebiotic. Oh, by the way, sometimes they got to throw all these terms. Synbiotic, S-Y-N, synbiotics is a new thing. I don't care that you really know that, but if you do see that, what it is it's usually sold as a pill, and it's a combination of some prebiotic and probiotics mixed into the pill. So it's the microbes plus the food that feeds them in one pill. And so then they'll label it as a synbiotic. So just be aware of that. And then there's this one. This is the newest term, postbiotics. Have you even heard of that one? I just heard about it the other day, and I read about this stuff all the time. So I don't, it's new. Anyways, of course, they got to throw more jargon. Oh, it's a metabolic, it's a biogen. What it is metabolite. I don't care that you know the first two, but a postbiotic is a metabolite that the microbes are making and excreting out. And then those metabolites can influence you. And it can also happen when the bacteria lyse open and die and their contents are released from their stuff. So it's just chemicals or metabolites that are being produced. And they have some kind of biological activity in the whole. So postbiotics, they're just the chemicals excreted by the microbes. I don't know why they had to throw yet another term at us, but anyways, I wanted to alert you to this because it's a thing that you'll probably hear more about in the near future. So here they're looking at it, food and pharmaceutical industry. So they're trying to, like the butyrate idea. That's what I mean. Like they're looking at, should we make pills that are filled with butyrate powder that you can swallow? Technically, that's a postbiotic. So this is something that you may see more about in the near future. Businesses popping up, starting to make this kind of thing and sell it. And if you really want to get wacky, yes. Could there like the butyrate idea? Yeah, I mean, I think any postbiotic that you take, it's going to impact your microbiome in some way. So they're going to have to do a lot of, hopefully they're going to do a lot of studies on that. And, the problem with supplements is it's not FDA regulated. And so if they don't do their due diligence and start selling that product, Yeah, I'd be a little leery. I would definitely want to do my own research on something like that before I take it. Like I think I may have mentioned I take probiotics, but I did a lot of research on that before I chose to do that, just to try to limit any downsides, right? Probiotics aren't regulated by the FDA, so you're kind of it's the wild, wild west. You have to do your own research on that. So, did I answer your question? Okay. So, um, wacky. Let's get really wacky. Epigenetics. So if you've taken genetics, this idea of epigenetics is, upon the DNA essentially, you're looking at ways of influencing gene expression without causing any changes in the DNA itself. You're not manipulating the bases, doing anything like that. There are other ways to influence gene expression. They're looking at the microbes now. How are they influencing gene expression of your own cell, your eukaryotic cell? And they're finding that done. Paper that was published, the link between early nutrition, microbiome, and long-term health development. And this is looking at human, um, moms who have children, babies, and following them out over the first 1,000 days, see what was happening. And so they're starting to come to the conclusion that the microbiome is an epigenetic modulator. And this, it gets really, really interesting and kind of wacky to think about. But basically, microbes in your gut have a way to influence your gene expression. And they're doing a lot of this with the epithelial cells that line your intestinal tract, looking at altered gene expression that way. That's a little easier to think about because microbes are right there. They're close to the epithelial cells. The microbes release chemicals. Your cells can take those up, and some of those can influence, can be an epigenetic modulator. But they're also saying that it can happen systemic. And so it gets very, epigenetics is just wacky to me. It's so cool to think about, but I still don't have, I'm not an expert. But so this is a figure from that paper. Looking at the first 1,000 days of life is where that's kind of the time frame that they said this is really, really important. So again, if you ever choose to have children, how you eat before you conceive, what you eat as you're pregnant, and then shortly after birth, breastfeeding, all of that, up to the first 1,000 days of your child's life is super important. That's what many, many studies are starting to support this idea. And they're saying, so this is trying to get at nutrition, both what the mom eats as well as what the babies eat when it's born, and then the microbiome in the mom as well as the baby, and then of course all the genetic component and where those three circles share, like we're saying, has influence here. So very interesting. Still a lot of work to go. They're coming up with some statements they can make, but there's still a ton of research that's needed on it. But again, I think it's just really, really important to choose to have children that you think about these types of ideas to, maximize your child's going to be strong and healthy and robust and good to go. So what we see, going back to the obesity idea and trying to minimize that outcome, whether it be a newborn or if you're already an adult, there's essentially two major mechanisms that they point to the microbes. The microbes have different abilities to extract energy from the food you eat. Some are really good at it, some are not so good. So if you want to stay away from the obese profile, you want microbes that aren't terribly efficient. You'd want ones that aren't that great at extracting energy from the food you eat. And they're starting to figure out which ones are not so efficient, which ones are a little too efficient. They're starting to look at that, and again, designing probiotics for that. So harvesting of energy from what you eat, and then this idea of what role do they play or don't play in inflammation in your body. And So one key thing about a certain group of microbes you should be aware of, lipopolysaccharides. We learned about them when we went way back, when we looked at gram-positive versus gram-negative cell walls, and we looked at that funky outer membrane, which is called the lipopolysaccharide of gram-negatives like E. coli. And what they're finding is, I mean, you have a bunch of E. coli in, and you have a bunch of other gram-negatives. But what they're finding is if it's imbalanced, where you get too many or some certain kinds of gram negatives as well, it can trigger inflammation. What they're finding is if you do eat a high fat diet, and again, going back to the ketogenic diet, which promotes you eat high fat, it can actually trigger inflammation. And the way of seeing that is as you eat high fat, you get a lot of gram negatives. Their populations increase in you. And remember the lipid A, especially the lipid A portion of the polysaccharide, that's considered an endotoxin. They're finding that it can start to cause leaky gut idea, where the endotoxin can leak past your epithelial cells of the lining of your gut and get into your blood. And that would cause full body inflammation. And so, again, high fat diet, even though there's a bunch of, diet gurus who say this is the way to go, ketogenic. From this perspective, I would say no, that's not the way to go, in my personal opinion. All right, so here they're looking at, okay, so what is the polysaccharide and the lipid A, the endotoxin? What is it really doing? So here, fat-rich is fat diet, you get a bunch of triglycerides that you're ingesting. They can form these vesicles, they can micelles, basically a vesicle. So LPS. So what they're saying is you can see that it can cross. So here are these three cells are your epithelial cells and your gut. And normally you have these tight junctions between your epithelial cells that provide secure binding of your neighboring cells. But with all of this going on, you can actually get those tight junctions breaking down, which is part of the leaky gut idea. So you can see that you have, they're proposing a couple of different routes for how the endotoxin could get past your epithelial cells and then into your bloodstream. Chronic exposure to endotoxin has been associated with obesity, diabetes, cardiovascular. So again, you have to think about those ideas and whether or not you want to continue to do, if you're on the ketogenic diet, I really urge you to think about this. Here's another one, intestinal permeability, high fat diet. For both barrier disrupting, but also it stimulates pro-inflammatory cascades. So it's kind of a double whammy. This is another study where they looked at the Western diet versus the more prudent diet, a healthier diet. They called the prudent diet. And then they were looking at how much LPS was in the blood and circulating through the body. So this was only done with 8 humans, but Western diet for 1 month induced a 71% increase in blood levels of endotoxin, whereas a more healthy diet reduced levels by 31%. So the Western diet might therefore contribute to endotoxins in the full body through the bloodstream. So here endotoxin activity, decrease go into the fruited diet, but actually increased in the Western diet. The Western diet is another acronym. Well, an acronym for the Western diet is SAD, SAD, Standard American Diet, which the acronym is SAD. I thought that was hilarious. So it pretty much is SAD, high fat, low. What am I trying to say? High fat, low carb, no complex carbs is what I'm trying to say. Too much sugar, too much fat. So inflammation and the lipopolysaccharide. Lots of studies being done in looking at how it can be a systemic issue. It doesn't just stay in the gut. And not just on obesity, but on cardiovascular, intestinal inflammation, low fiber diet, high alcohol consumption, high fat. All of this can contribute to leaky gut syndrome. I'm sure you might have heard of leaky gut at some point already, just from reading or hearing some of the news that gets taken up by the news media. So the issue is we have a lot of gram-negative bacteria in us. That's in and of itself, that's not the issue. It's when you get a dysbiosis, imbalance, or you get too many of them, then you can start to see these inflammatory responses in your body. Translocated LPS promotes chronic neuroinflammation by the production of brain tumor necrosis factor alpha. You don't need to know the details of that, but brain inflammation is something to think about. And so, like I said, systemic. Your brain is not immune to this. You can get these chemicals getting into the brain. So this is a figure trying to show this idea of what you eat or medication you might take is impacting the population ratio. And so here they're just showing like a teeter-totter idea of because of and they're trying to show that there were some poor choices made, which caused an increase in the Firmicutes phylum and a decrease in the Bacteroidetes. And so again, it's dysbiosis or imbalance that can lead to progression to obesity. Here's another one trying to get at the same idea where they're looking at the gram-positive versus gram-negatives in you. And again, if you get too many gram-negatives, I'm showing here the teeter-totter idea. And when the gram-negative went too high, and show the LPS into the bloodstream and across the epithelial, and resulting in inflammation and metabolic disease is getting at obesity, but it can also be other things too, diabetes and so forth. So we're going back to this idea. All right, this is the gram-negative cell wall structure. It's a lipopolysaccharide, in particular. So again, if you get too many of these, where it can trigger inflammation. So this is a book written by Dr. Paul Clayton, who came here to Northern a few years ago and gave some seminars on this idea of your microbiome. And he studies it historically, looking at pre-industrialized population through current, and he was coming to the same conclusion that inflammation is a huge reason for some of the diseases we currently see in the human population. So that chronic inflammation. Yeah. Here, they're actually trying to use easier to work in non. Here they were looking at broiler chickens primarily because they want to sell these for food. Over in Europe, this is a paper published over in Russia. Over in Europe, they've outlawed giving antibiotics to animals you raise for food. I couldn't believe it. They did it in 2020. I was very happy, but we haven't done it over here. We still allow antibiotics in food production. It's terrible. In fact, that's where most of our antibiotics go. They pump the beef cattle and probably the milk, all the milk cows too, and all the chickens and everything we eat, all the meat products. They're typically been fed a lot of antibiotics. Do you know why? The obvious is disease prevention, right? And that does do that, but they figured out before they started using it this was before they started packing all the animals so tightly together. So we're talking quite a while ago. They figured out if you give low doses of antibiotics to cattle or chickens, they put on more meat more quickly. And they actually found this out in the military. They were giving soldiers low dose antibiotics, and they found that those soldiers could bulk up and increase their muscle mass much quicker. And so then they took it into, well, could we do that for meat production? And so that's really what kind of triggered antibiotic production or antibiotic use in meat production. And then they were like, well, we can use it for disease, trying to knock out pathogens too, especially when they started putting all the animals together, really closely together. But anyways, with this, they're looking at corn sensing. So over in Europe, they're forced to do something else. They can't use antibiotics now. And so they're looking at corn sensing to reduce inflammation. So again, this idea of inflammation being caused by certain microbes. And in this case, you can't give the chickens antibiotics to help them get rid of those microbes causing the inflammation. So if you can somehow use the corn sensing inhibitors to stop those microbes from communicating, then hopefully they'll ramp down and the inflammation will go down and you'll be able to sell those chickens. And so the quercetin, vanillin, and umbelliferon, those are not anything dangerous for us to be exposed to. Quercetin is actually healthy for you, and vanillin too. I don't know much about the third one, but I know the first two are healthy for you. So anyways, looking at how to reduce inflammation again. Oh, I have to flip to the other slide set. Is the other slide? Oh, we're out of time. Darn it. All right, so on Friday, I'll get into the second slide set. I'll probably wrap up this idea of your microbiome